Thanks, Uta, for the lovely introduction. I am absolutely delighted to be here. I've had such a wonderful day, and so I just first want to say thank you to everybody for sharing their spectacular science with me. I don't know when I've learned so much in such a short time, so it's really been fun and heady. And I'm also grateful for the opportunity to tell you a little bit about some of the projects that we're doing back in Princeton. And so I'm just going to tell you two brand new stories. And so to start with that, I should just tell you, and Uta already hinted at this, that my lab works on how bacteria communicate and how they control collective behaviors. So traditionally, bacteria get a lot of bad press because there's all these bacteria that are in the world, in the environment, that have no business being in or on a human or a plant or an animal. And if they can get a toehold, they can make these higher organisms sick, or they can kill them. Increasingly, in the last decade or so, what my field is starting to learn is that beyond doing harmful things, we get now that bacteria have actually given us life itself. Because all higher organisms live in a consortium with mixtures of bacteria that provide us biochemistry and products that our own genomes don't have. And so we're only now learning about all of these beneficial traits that bacteria have, but just a few that we know about. For example, we don't have the enzymes to digest plant products, so bacteria give you access to those calories. They provide us with micronutrients, and what we now know is that they work hand in hand with our immune systems to keep invaders out. So traditionally, we've known about harmful bacteria. Increasingly, we're learning about beneficial bacteria. But the question that is at the heart of my lab's work is whether you want to think about all these bad things bacteria do, or you want to think about all these magical things bacteria do, the question my lab wonders is, how do they manage it? They're so small. So how do bacteria get any bang for their buck? And what I want to try to show you today is that the way they accomplish these feats is by acting as groups, coordinating their behavior, and carrying out tasks that they could never be successful at if they acted as individuals because they're too small. And so that process is called quorum sensing. And so in the sort of simplest cartoon way that we think about how bacteria do this is sort of shown on this slide. So at low cell density, when bacteria are alone, they want to have the program of gene expression going that's good for acting as an individual. So they're carrying out some subset of the tasks that they can do, and among the things they're doing is they're making and releasing small molecules that we call autoinducers. And I have these as the red triangles on this slide. So the world is big and the bacteria are small, so at low cell density, the autoinducers diffuse away, the bacteria can't detect the autoinducers, and that says act as an individual. But then as the bacteria grow and divide, since everybody in the environment is making a share of the autoinducer, the extracellular concentration of the autoinducer increases in proportion to cell number. And when the molecule hits a particular threshold concentration, the bacteria detect it, and they infer from that detection event that they must have neighbors around. And so they're actually using the concentration of the autoinducer as a proxy for cell number. So when they detect the autoinducer, they say, okay, I have neighbors around, and then in unison, all of the bacteria change their gene expression, which translates to changes in behavior in synchrony. And they begin to carry out behaviors that are only successful when they do them as groups. And so I have this bacteria drawn in blue because originally quorum sensing was discovered in bioluminescent marine bacteria, and so we could just see them turn on light. But now what we get is that quorum sensing is the norm in the bacterial world. There are hundreds and hundreds of cases of bacteria that we know have quorum sensing systems. Typically, they'll control something like 200 to 600 genes by these circuits. And the kinds of behaviors that they control are things like biofilms, which are how bacteria sit on surfaces as communities, and the group production of virulence factors. So what we get now is that if bacteria are mutant for quorum sensing, they can't form biofilms and they're not virulent, right? And so those are sort of the big things that people have been studying in the quorum sensing field, but I think the way you need to think about this is about public goods. Whenever a bacterium is going to give something away to the world, it never gets its own product back. So only when these kinds of public production of goods happen in groups do they become successful, because you get mine and I get yours, and then the entire consortium succeeds, right? So it's really about public goods and exoproducts. So that's quorum sensing in a cartoon. These Vibrios are the model for quorum sensing. We still use them as models, and I'm only going to talk to you about Vibrios today. And so I'm going to show you the architecture and the molecules from some Vibrio circuits. And so what the bacteria do is they fiddle with how many molecules they make. They fiddle with what the components in the circuitry is. But the principles that I'm going to tell you are conserved in all quorum sensing bacteria, even if the components differ, right? So the rules are the same. Okay, so starting with these Vibrios, what my lab showed is that there must be at least 3 molecules in this language that allows bacteria to communicate and coordinate behaviors. So in every species of bacteria that's been studying, there's some molecule, and in the case of this Vibrio, this molecule, which is a homoserine lactone, there's some molecule that that bacterium and only that bacterium makes and then detects with the partner receptor. So this molecule is exquisitely specific. It confers intra-species communication. This is how I count how many of my sisters and brothers are around. There's a molecule that we discovered that all Vibrios make, but from genome sequencing and sampling projects, we can see that apparently no other species of bacteria has the capacity to make this molecule. So we think this is the Vibrio language. This is for intragenous communication. So this says you're my clone, my sister. This says you're my cousin. You're a Vibrio. And then there's a molecule that we discovered that is broadly made in the bacterial world, both by gram-negatives and gram-positives, and all the bacteria make exactly the identical molecule. So this molecule doesn't have species information in it. It just says, I'm a bacterium. So this is for interspecies communication. So what we now think about quorum sensing is that these molecules encode a lot of information. They certainly encode something about cell number, but what they also encode is something about how closely or distantly my neighbor is related to me. And what we've recently shown the bacteria do is that they actually measure the ratio of these molecules. And I think what they're trying to ask, is it me and my kin that are here, or is it me and the enemy that are around? And what we know they do is, based on the blend, the ratio of these molecules that they perceive, they actually carry out different tasks, whether they and their siblings are in the majority or somebody else is. So they can distinguish self from other, and they act appropriately. Okay, and so again, different bacteria use different blends of molecules, and there can be more or fewer molecules in the conversation, but there are at least these 3 kinds of molecules. Okay, so that's the molecules, and now I'm going to talk about how the bacteria send the information that's contained in the molecules into the cell to control gene expression. So at low cell density, when the bacteria are alone and the molecules aren't there, these 3 cognate receptors that sit on the cell surface, they are kinases. So in the absence of the autoinducers, these receptors autophosphorylate, and they send phosphate to this protein LuxU, which then transfers it to this protein called Luxo. So Luxo is a transcription factor. So at low cell density, it's phosphorylated, and then it turns on the expression of a set of small RNAs. And for those of you who work on eukaryotes, these are microRNAs. This is the bacterial version of microRNAs. So at low cell density, these small RNAs get made, and then what they do is that they act post-transcriptionally. So the small RNAs sit on the messenger RNA encoding this protein AFA. And when they sit on that messenger RNA, AFA gets translated. So this protein AFA gets made, and that's good because it is the master transcription factor that runs the individual cell program. The small RNAs also act negatively, and so in that case, they sit on the messenger RNA encoding this protein HAPR, and the RNA gets destroyed. So HAPR doesn't get made. And that's important because this is the protein that's the master transcription factor for group behaviors. So at low cell density, AFA gets made, and the bacteria commit to being individuals. Then what happens is, as the bacteria grow and divide and these autoinducers accumulate in the extracellular environment, when they get bound directly by the receptors, so the receptors actually bind those autoinducers, that binding event flips a switch. And the receptor changed from being kinases to being phosphatases. So now the phosphate flow reverses in the circuit. When Luxo is dephosphorylated, it's inactive. the small RNAs don't get made, so now that means this AFA protein doesn't get made, but HAPR isn't repressed. So at high cell density, this protein HAPR gets made, and it turns on the 600 genes that are involved in group behaviors. So it's actually really simple how this works. Either a low cell density transcription factor is made or a high cell density transcription factor is made, and that depends on whether or not these molecules are there, and that depends on whether or not the bacteria are alone or in a group. So that's how the circuit works. And so my lab, the gang in my lab discovered everything that you see in this slide. And we actually study, as Ute said, we study all of the layers of the circuit. We're interested in finding molecules and understanding what is the information encoded in them. We try to understand how do the receptors detect those molecules, and how do they distinguish between other closely related molecules so the bacteria don't get confused. We try to understand how the circuit works, and then what we really want to know is what does it mean for a bacterium to act as an individual or to be a member of a group. Right? And so today, to just try to give you a flavor of the kinds of projects that we do in the lab, as I already told you, I'm going to tell you just 2 stories that are really new. And so one will be about molecules and the circuit, and then the second story that I'll tell you is about a behavior, a quorum sensing controlled behavior. So everything that I'll tell you about today is in the pathogen Vibrio cholera. And so Vibrio cholera, as far as we can tell, only uses 2 of these molecules. And so, of course, you know about Vibrio cholera because it's a global pathogen. You get cholera from drinking contaminated food or water, right? And it gives this terrible diarrhea. It causes an acute disease. And from the point of view of cholera is it gets in, it makes you sick, and then it gets out to infect the next person. And so what my lab showed is that the bacteria need quorum sensing to be a pathogen. And so the way that it works in cholera is that at low cell density, as I told you, when the autoinducers aren't there and the phosphorylation cascade is working the way I told you, and this protein AFA gets made, it turns on adhesion, so how the bacteria make a biofilm on the intestine, and it turns on the expression of all of the virulence factors that make a person sick. Then when the autoinducers kick in and the system reverses and HAPR gets made, its job is to shut off all of the virulence factors, all of the biofilm formation genes, and it turns on a set of genes that lets cholera cut itself off the intestinal epithelium, and out it comes by the gazillions. So again, the bacteria have to have quorum sensing to be a pathogen, but the circuit is poised for dissemination. And so the insidious strategy is for cholera to get in. It comes in with guns loaded, right? It's got its virulence factors on it, makes a biofilm, and then before you know it, it's multiplied like crazy, and then it leaves, and it does this, it gets back into the environment, and that's how you get these endemic diseases, right? So if you're listening to the logic of the circuit, autoinducers or pro-quorum sensing molecules flip cholera into high cell density mode, which means don't be a pathogen. Right? So autoinducers repress pathogenicity. So we had figured out the parts of the circuit, and what's wonderful about all quorum sensing systems is that they come with these molecules, right? And so you actually can we can make all these molecules in a test tube, right? You can turn the system on demand. But what nature didn't give us were molecules of different potencies. Nature didn't give us antagonists of the receptor, and nature didn't give us molecules that work from the cytoplasm. And so what my gang, Amanda Hurley, a graduate student, wanted to do was to sort of expand the repertoire of molecules that we had to work with to use as probes, and then also to think about therapeutics. So we wanted probes to understand how the circuit works, and then we want, this is a pathogen, so we wanted to try to think about this in medical terms. So what Amanda did was she carried out a screen of synthetic library, of just synthetic molecules, and she looked for agonists of the receptor, and she got them, and she looked for antagonists of the receptor, and she got those too, and we've studied those a lot, but I'm only going to tell you about one molecule today because what she got was something that was completely different from what the system normally comes with. She got a molecule that is a pro-quorum sensing molecule that works from the cytoplasm. So what Amanda got is a molecule that we named AZU, and so this is the structure of the molecule, and it is an inhibitor of this protein Luxo. And you have to remember, Luxo is a negative regulator. So inhibition of Luxo turns cholera into dispersal mode. And so we wanted to study that molecule and how it inhibited Luxo. And in order for me to tell you about that, first I have to tell you about Luxo. So Luxo is what's called a AAA plus ATPase, and these are broadly spread in prokaryotes and eukaryotes. Like if you work on eukaryotes, dynein is a AAA plus ATPase. And so they all work sort of the same way. They're regulated by something. In our case, they're regulated by phosphorylation from the receptor. Then they hydrolyze ATP in order to get energy to do something. In our case, it's to get energy to melt the DNA so that transcription can happen. So the way Luxo works, if you remember the circuit, is that low cell density, it gets phosphorylated through that cascade, so that activates the protein, it hydrolyzes ATP, and then that melts the DNA, and RNA polymerase can get in here, and then the cells make those small RNAs, which are the regulators. Then at high cell density, Luxo is dephosphorylated, so it's not an ATPase, and so no transcription happens. So that we knew about Luxo by the kind of protein that it was. And so somehow, this molecule that Amanda found, AZU, changes Luxo from the active low cell density form to the inactive high cell density form. And we wanted to know how. And so what we decided to do to try to understand mechanistically how that was happening was we took a structural biology approach, because what we realized is that even without AZU, we don't know how Luxo, other than this cartoon, goes from this to this anyway. So what we thought we would do is we would crystallize the protein, and we did that 3 times. The apoprotein, the ATP-bound protein, and the AZU-bound protein. And so we thought if we compared the 3 structures, we could learn about natural regulation of Luxo and then synthetic regulation with this quorum sensing, pro-quorum sensing molecule. So as I told you, we crystallized it. The protein crystallizes as a hexamer in a ring, and that is very common for AAA plus ATPases. So now what I'll do is I'll zoom in, and I'm going to show you a monomer, and I'm going to show it to you 3 times. So first, the apoprotein. So this is the unphosphorylated inactive protein. So what we know is that this is where the protein gets phosphorylated by the receptors. And this is where ATP binds. And so the protein has 2 domains. It has a regulatory domain that interacts with the receptor, and then it has this ATPase catalytic domain. And so instantly, when we got this structure, we actually understood how the protein naturally keeps itself off in the absence of phosphorylation. So what I've drawn in yellow is that linking the regulatory domain to the catalytic domain is this yellow sort of finger. And when you look at the crystal structure, that yellow linker domain that attaches that linker region that attaches the 2 domains, it sits right in the active site for where ATP should go. So it's actually like a competitive inhibitor. So in the off state, the protein is inhibited because there's a few amino acids attached to the protein sticking in where ATP should bind. So that was the first clue. And then consistent with this, remember I told you we crystallized the active protein. So now we crystallize the phosphorylated form of the protein with ATP bound at the active site. And what happens is when the protein gets phosphorylated due to quorum sensing, this linker stretches out, making room for ATP to bind, and so now ATP is sitting there, and it gets hydrolyzed, and transcription happens. And the reason that we're especially interested in that is because there are tons of AAA plus ATPases. I already told you that. They all have to be regulated. I already told you that too. There are lots of crystal structures. Never so far has there been this little competitive intramolecular linker domain. So this seems to be a new mechanism that this protein Luxo uses to keep itself off that's different from every other known AAA plus ATPase. So then we crystallized it with the inhibitor bound, and the inhibitor, AZU, was sitting right in the ligand binding site. And of course, we did companion biochemistry, and indeed, this molecule is a competitive inhibitor of ATP hydrolysis. So the protein works by competition, and the inhibitor works by competition as well. And so what's fun about this inhibitor is if I put the first protein structure back, this AZU molecule actually traces out the natural path that linker makes. It actually makes all the same hydrogen bonds. And so it's simply taking advantage of the natural mechanism of inhibition that evolution gave us. Right? So now we have this protein and we have this inhibitor, and we understand a lot about how these 2 things work. But if I go back to my circuit, what you'll also remember that I told you is that an inhibitor of Luxo should flip cholera into high cell density non-virulence mode. right? And so we did all of that crystallography to understand how AZU works, and we got the mechanism. But the real question is, put your money where your mouth is, does it actually work as it's a pro-quorum sensing molecule, but isn't an anti-virulence molecule? And so what we did was we measured production of the major virulence factor, which is called TCPA, which is toxin co-regulated pilus. So when cholera gets into the intestine, it adheres to the intestine by this protein. So that's the first virulence factor that it makes that allows cholera to adhere to the intestine, and then it secretes cholera toxin. And so what you can see is this is all at low cell density. Cholera makes a lot of this virulence factor. But as we add increasing amounts of the AZU inhibitor, sure enough, we can shut it down. And so indeed, this molecule that inhibits Luxo will turn off virulence in cholera. And so we're interested in that because of cholera, but we're actually more interested in that when we actually step back and think about pathogenic Vibrios in general. So I presume that you've heard about Vibrio cholera because it causes a pandemic disease. But there are many pathogenic Vibrios on Earth, and they kill a lot of people, primarily children, right? Cholera is the biggie. But these other pathogens, as well as other Vibrios, are also pathogenic. And so what's interesting from my point of view with respect to quorum sensing is you'll remember that I told you that bacteria have to each interpret their unique blend of autoinducers. So they tinker with the tops of these cascades. So there's a lot of energy in trying to make these anti-quorum sensing or quorum sensing manipulation strategies to think about pathogenicity. And so people, including me, try to make them up here, but those tend to be very species specific because each species of bacteria has a particular blend of molecules. Likewise, there's a lot of activity in the bacterial field now to try to wonder, can you turn off a particular virulence factor and that's enough to stop pathogenicity? So lots of people are working on different kinds of virulence programs. So each of these bacteria has a unique quorum sensing blend. They always control virulence in Vibrios, but each bacterium deploys a particular virulence program, and that causes the specific disease. What's fantastic about the way Vibrios are set up is all Vibrios have this stuff in the middle. That is in every Vibrio, and it's nowhere else. So Luxo is a Vibrio protein, but as far as we can tell, no other species of bacteria has Luxo. And now I told you that Luxo has this unique inhibitory mechanism, right? So what we did was we took that AZU molecule and we tried it on all of these pathogenic Vibrios and lots of others, and sure enough, it shuts down pathogenicity in every Vibrio. So on the one hand, the molecule is really broad spectrum. It's an anti-virulence molecule for Vibrios. But on the other hand, the molecule is exquisitely specific because it only hits Luxo. And so we have tested it on lots of other bacterial and eukaryotic AAA ATPases, and it doesn't work. And that, I think, is because of the way the molecule works with this business of this linker where it exploits this new mechanism of natural inhibition. So we're really excited about this molecule because we think we have a broad spectrum, narrow spectrum, anti-virulence molecule for Vibrios. And so what we're doing now that we think that there's merit to this idea is we're now getting really serious about doing the medicinal chemistry, right, to make that lead molecule that I showed you look like a medicine and get where we want it to go. And so we are happy about that, and we're going forward. And I think the thing that's sort of underneath this talk is the big question, which I think AZU is going to let us test, is can you stop an infection by interfering with behavior and not interfering with growth, right? This molecule is not bacteriostatic, it's not bactericidal, right? And if we or somebody could do it, you can imagine a whole new way to think about antimicrobials, right, that might not be as prone to resistance as traditional antimicrobials. And so that's where we're going with that story, and we're working on the chemistry now. Okay, so that's my first story, which has to do with molecules and the receptors, and now I actually want to talk about quorum sensing and what it means to switch between these two behaviors. And so the behavior that I'm going to tell you about today is biofilm formation. And again, it's in Vibrio cholera. And so I know that the Karolinska is a powerhouse in biofilm formation, but just for those of you who aren't in those labs, I want to remind you about this behavior. So what we now get what we get is bacteria do not live shaking around in flasks. It turns out that in nature, they live in biofilms. So they live predominantly adhered to biotic and abiotic surfaces, right? So this is the environmentally and medically relevant context for bacteria. And so we know some of the steps in biofilm formation. So what we know is that the bacteria are modal, and so they will swim to a surface and they attach. And then they begin to secrete a matrix, which is made up of polysaccharides, proteins, and DNA, and that encases the bacteria. It adheres them to the surface, and it adheres them one to another. So it's like a suit of armor. And so they begin growing in this matrix, and then they mature into these large communities of cells, all embedded in this matrix. And then at a particular time, some cells in the biofilm will regrow their flagella, and they leave the biofilm, they disperse, presumably to find new real estate, and they will do this over and over again. And so when I look at this picture, I mean, it's a cartoon, right? What is sort of especially interesting to me is that for bacteria to be in a biofilm, it means they're adhered to the surface. So that means they're in 2 dimensions. But all of the nutritious products, all of the stuff the bacteria need to live and grow is flowing by them in the 3rd dimension. So the sort of first question, just a basic question, is how can the bacteria be 2-dimensional and 3-dimensional at the same time, which they have to be. They have to be stuck to the surface, but they have to access the food. And so I'd like to understand how they form these films, right? And the problem, which has been in this field since its inception, right, is that these are opaque. Right? So for those of you who work on eukaryotes, bacteria are 500 times smaller than a eukaryotic cell, so they are intrinsically hard to image, and they're really hard to image when they're in these communities. And so the problem is that my field has this cartoon. We know these biofilms are important. We know that they're living in these communities, but in fact, we don't have a clue about the steps because nobody has imaged them past this step. Right? And so what I should also tell you is that Quorum Sensing controls these transitions, so that's why I'm interested in it. Right? And so my lab really wanted to get out of the flask and start studying these bacteria and studying how Quorum Sensing plays out in environmentally and medically relevant contexts, right? So we wanted to take this on. But in order for us to be able to learn something about how Quorum Sensing is playing out and what different jobs, different cells inside these films are doing, we had to be able to crack this problem and image these cells. So a postdoc in the lab, Jing Yan, who's a physicist, basically said to me, Bonnie, this is an optics problem. Let's just solve it, he said. And so he did. So what he did was he built a custom confocal microscope in which every button on the microscope is for imaging tiny objects. He reduced the phototoxicity, because mostly what happens is you kill the cells when you try to image them. And then he also wrote these custom algorithms that allowed him to segment the cells and tell who was who. And so now I will show you what he got. What we can do now is we can watch these biofilms develop cell by cell from the founder cell all the way up to about 10 or 15,000 cells. Okay, so this is Vibrio cholerae. It's got a fluorescent reporter in it. This is the founder cell, and I'm showing you the bottommost slice from the confocal microscope. And so then you're going to watch over time. It divides. It spreads out radially. It makes this like pancake-like base of the structure. Right? So that's the first slice, and so what Jing does is he does that, but he makes 150 slices through the biofilm, imaging the cells in the time of them dividing, right? And so then what he can do is he can put all of these images of each slice together, and then cell by cell, he can build the biofilm. So here you're looking down, and here you're looking from the side. That's supposed to look like that cartoon. And so what you can see now is every single cell on the periphery of that biofilm, but from bottom to top. But what I can tell you is how every single cell inside that biofilm is arranged. I can tell you how close or how far it is from its neighbor cell, and I can also tell you who its mother was, who its grandmother was, and who its great-grandmother was. So he's got it, and we can watch these films develop, and by doing that, we can actually put steps into that cartoon for how this happens and make a model. And so indeed, as was known, the bacteria cholera swims down to the surface, and then they adhere, and they always adhere on their long axis. And so now they start to divide, right? But they begin to produce this matrix, right? But they are adhered to the surface. Remember that. So then they keep dividing. And now what happens is actually really simple. What happens is they're stuck to the surface, but they're trying to divide. So every time a cell divides, it's got to make room for its daughter cell, but they're stuck to the surface. So there's a problem, which is there's a force holding the cells to the surface from the matrix, and then there's a force of the cells in the middle pushing outward. And what happens then is at a particular moment, the force to divide exceeds the force holding the cells to the surface. So right in the middle of the biofilm, all the cells stand up on end, and now they solve the problem because now they're in 3 dimensions, right? So it's simple. It's not fancy. It's just using compression forces. They stand up in the middle, and then cholera divides from the poles. So every baby cell that's born after that is born from the pole and automatically extends further into the third dimension, and so that goes on, and then you get this hemisphere that I showed you the real picture of, and in my cartoon I just have 4 layers, but that biofilm I showed you had about 16 layers in it, and now we understand how the cells are arranged. They are not willy-nilly, they are packed pneumatically, they are one next to another like soldiers in a tower, right? They're all standing straight together, and they are they're remarkably organized, and they are remarkably close together, and then on the edge they're sort of decorated by these cells that are radially organized on the periphery. Okay, so now we know the steps, right? And I've already told you that quorum sensing controls this, and so what's fantastic from my point of view is there are only 3 proteins involved in that matrix, right? So it's not like a eukaryotic cell. There's only 3 proteins that are controlled by quorum sensing that make the matrix, and we're geneticists, so we can just knock them out. So what's even better is 2 of the proteins act redundantly, right? So there's 2 proteins that are called BAP1 and RBMC, and they are secreted by the bottommost cell. They're the adhesion that sticks the cholera to the surface. So Jing made a double mutant that doesn't make that glue and asked, using the same technology, what happens. And so here I'll show you the movie. And so what you can see is that the cells start dividing, but they're not organized, they're not sitting down on the surface, they're still stuck together, right, by polysaccharide and that 3rd protein, right, but they're kind of disorganized, and they're just sitting on remember, this is the bottom slice. And so we can stack up all the slices, and in my cartoon what we find is that the mutant that doesn't have the adhesion, it makes a sphere. So instead of the hemisphere, right? And so, and they're disorganized inside of it. And so that is the outcome. And then of course we can try to ask questions about like, well, why is that bad? I mean, if you're following what I'm telling you, this is still a 3-dimensional object. The nutrients are still flowing by. The bacteria have access to that, right? So the question is, what's wrong with that structure? And there can be lots of things, but the thing that we thought about has to do with resilience. So of course you guys in this institute know that the reason biofilms are such a scourge in medicine is because they are adhered to surfaces and you can't get rid of them. This one's not adhered to a surface, and so we just thought, well, maybe that makes it fragile if we add flow. And so we do all of this in microfluidic devices, and so what Jing did was he simply added a little bit of flow and asked what happens to the wild type and to the mutant biofilm. And so what we see, this is the wild type, that if you add flow, that's 100%, the cells just stay there. If you make either of those single mutants, the cells just stay there. But if you make the double mutant and you just add flow, they can't resist it. Right? So they're washed off the surface, and so of course you're not a biofilm if you're not stuck to the surface, right? So presumably that's one of the reasons the bacteria have evolved these proteins. Okay, so that's 2 of the 3. The 3rd protein is called RBMA, and its job is to stick sister cells together. And so Jing did the same trick. He just made a mutant in RBMA and asked, what does the biofilm look like? And so here you're watching the bottom slice again, and so what you can see is they stay adhered to the surface. They should, but what you might remember from the first movie is that in fact they make a much bigger base, right? And of course the intercell distance between the little yellow cells is bigger because they're not stuck one to another. Okay, so again we can look at these both from the top and from the side. And so what we see then is that in the wild type, now you're looking from the side, right? We get this beautiful hemisphere where the cells are packed really tightly. In the RBMA mutant, it makes a huge biofilm, right? But the distance between the cells is much bigger. And that's just all filled in by polysaccharides. Okay, so I can make my cartoons. Here's the wild type, here's the mutant. Okay, so now what's the problem this time, right? So it's adhered to the surface, so it's still a biofilm, right? And it actually can explore more of 3 dimensions because it makes a bigger biofilm. And so the question that Jing wondered is, well, why is that bad? And so what we thought about, now it goes back to quorum sensing and public goods. So what you'll remember is that all of these biofilms that I'm showing you start from a founder cell. So every cell inside of them is a clone. Right? And so what we wonder is maybe by packing so tightly, they're trying that they resist invasion. Because remember, quorum sensing is happening in here, and they're making all these public goods that everybody is sharing in the profits. Maybe you only want those for your kin, right? And you don't want to share, you want those to be privatized, not shared. And so what we did was we tried to do a little invasion experiment. So what we did was we added isogenic cells to each of these biofilms and asked what happens. We just colored them with different fluorescent reporters. And now what you have, so we added them to the wild type, and what you're going to see is that they can't get in. So the colors are reversed in this one. So the red fluorescent cells are the cells that made the biofilm. So here you're looking down, here you're looking from the side, and then we added isogenic wild type yellow cells, and what you can see is they can't get in. When we do it with this mutant, right, we add the cells, and what you can see is they get right in, right? So now the founder cells are blue. Here's the biofilm from the top, from the side, and what you can see is that the yellow invaders get right in, and presumably they're gobbling up all of the goodies that these cells are making as a collective. And so there can be a lot of other reasons why this is not a good solution, but what we think that this does, these are like the tight junctions, right, in eukaryotic cells. By lining the cells up really close together, right, nobody can sneak in and get all the goodies. Okay, so that's sort of the beginning. We've just built this technology, and I'm excited about it because it gives us access to these incredibly relevant structures, right, that we've never been able to ask these mechanistic questions about before. And so just to finish up, let me tell you, what we're going to do now that we have this microscope. And let me tell you, anybody can have this microscope, right? And the stuff, all the stuff is out there, the algorithms, all of it, right? But the things that we can imagine, right, which are so like goofy, you can't imagine these experiments haven't been done, given that there are thousands and thousands of papers on biofilms, right? It's asking so simple, like what genes turn on in different regions. Like what you can imagine, right, is that the cells on the outside or the bottom, that their lifestyle is different from the cells in the middle, right? And so finally we can ask what jobs are each of these cells carrying out. So now what we've just done is add, fluorescent transcriptional reporters to genes of interest, like quorum sensing genes, right? And we watch these films develop under the microscope and ask who turns on what gene and when, right? And so we can do that one by one, and we are doing that with key genes that we think should be important. But the other technology that a new graduate student in the lab, Justin Silpe, worked out is to actually ask that on a genome-wide scale. So what he developed is a technology that's called photo conversion. And so what one does is one puts a fluorescent reporter into, say, cholera, and it's green. But then when you shine light on that fluorescent, on the cells that are green, they convert to red. And so you can shine the light regionally. So for example, we can shine it on the bottom, or we can shine it in the middle, or we can shine it on the top. And then what we do is we just fix the cells, scrape them up, and throw them in the FACS sorter, and we sort the green ones from the red ones, and you do RNA-seq. So now on a genome-wide scale, you can ask regionally what genes are turned on where, and you could actually take those RNA-seq profiles and compare them one to another and get a global genome-wide understanding, hopefully, of what genes turn on when and where. Here's just to show you the technology. This is a two-dimensional biofilm, right? All the cells are green, and he zapped them with the laser right there, and we can, convert just a couple of cells if you care to do that, right? So that's letting us do it on genome-wide scale. And then the last thing, so both of those are passive. We just let the bacteria tell us what they're doing. The real dream in this field is to be able to perturb these films on demand. So the last thing that we've done is we've built optogenetic reporters into cholera, right? So you shine light and then genes turn on and off, right? And so what's so wonderful, I think, about our future and sort of the horizon for us is like we could, for example, shine the light into the middle and say, make it one particular autoinducer. Or we could tell a guy inside, regrow your flagella and disperse, or we can mislocalize where they make that glue. And we can ask what happens to that cell, what happens to its neighbors, and then also what happens to the global architecture of the entire film. And so finally, I think we're in a position now to perturb these on demand and to actually learn, for example, why, what is going on that makes cells in a biofilm 1,000 times more resistant to antibiotics than isogenic planktonic cells, right? And so by having these gene expression profiles and tricking the bacteria and doing the wrong thing at the wrong time, I think we're actually going to learn about that. So that's where we're going with this new microscope that, that we just made in the lab. And then when I step back and go back to my circuit, remember, of course, we're going to exploit all these mutants and molecules in order to, in combination with that technology. But again, this business of not studying these bacteria in isolation in a flask, I think that's my labs, and I hope the future of this field, right? Like in 20 years, all these quorum sensing labs, we've learned a lot about these molecules and how these circuits work. And so I actually believe now that as like in these 3D heterogeneous biofilms, we can actually learn something meaningful. But I actually think we know enough now in my field to learn something meaningful about bacteria living in mixtures of species, which is how they live outside of the lab. And so the last thing that I'll tell you that we're trying to do, because we have a really interesting, I think, new finding here, right? When cholera is a pathogen, right, all of this business of quorum sensing that I told you about plays out in the context of the intestine. So there are eukaryotic cells, and there are also hundreds of species of resident commensal bacteria, right? And so the question is, how does this work to make this bacterium be a pathogen in the context of all of these other species, right? So now my colleagues that are in the microbiome field are doing sampling experiments and sequencing experiments, and we know that many of these species have the quorum sensing genes that we know about. So clearly, quorum sensing is going on here, and if you take samples, you can find those molecules that I showed you. So those molecules are being made in the intestine, right? And so the question is, how does that play out in health, and how does that play out in the context of disease? We like cholera a lot, and so the last thing that I'll tell you is that a former postdoc in the lab, Kai Pappenfort, he wondered about this, and he wanted to take the first steps to sort of up the complexity and begin to study this in the context of the microbiome. So what he found was that the microbiome cells make this brand new molecule to biology, which we call DPO. But what we know is that microbiome cells, one of the substrates they live on is mucin. So that's the mucus that covers your intestinal cells. It's made of 35% threonine, and if you look at this half of the molecule, that should look a lot like threonine to you. And so in fact, the microbiome cells use a host-produced substrate, mucin, to make this molecule, which cholera interprets as an autoinducer. And what you'll remember from my whole talk is that autoinducers make cholera disperse. So now we have this fantastic three-way interkingdom partnership. The host provides the substrate, the microbiome, the commensals, make the autoinducer, and the pathogen detects it. And in this case, it limits its infectivity. And so indeed, if you take commensal bacteria that don't have mucinases, cholera will make that mouse much more sick than if you have commensal bacteria that can actually make this autoinducer. And so anyway, I think that now, these are really complicated problems with hundreds of species, 3D and all this stuff going on, but I think that there's a way now to start thinking about these molecules and actually using the stuff that my field has learned over these past couple of decades to begin to tease apart at a mechanistic and molecular level at least some of these interactions. And so anyway, this is where we're going. He's figured out the entire circuit for how that, this molecule, there's a new receptor, and then it controls biofilm genes, but it tells cholera to turn them off, right? And so we're trying now to actually put together this sort of reductionist stuff with these much more complicated circuits and begin to move into this 3D mixed species interkingdom world. And so you can invite me back in 20 years. I come here every 20 years, right? And I will tell you, I better give an update. And so now let me finish. This is my gang. So starting from, this is Kai. This is his party because he became a professor in Germany. So this is Kai, who found that new autoinducer. This is Amanda, the graduate student who did the crystal structure and found that AZU molecule. And then this is Jing. He's the physicist that built the microscope. And then for all of the students, he also made all of the mutants, all the reporters. And so like there's a way to do it now where you put a foot in these two different worlds, right? And you can, you know, come as a physicist and leave as a biologist, or come as a biologist and leave as a chemist or a physicist. And so all of this gang is really into this sort of mixed up chemistry, biology, physics sort of science. And so I want to thank Uta for inviting me. I'm really having just a lovely day, and I thank you all for listening to me, and I'm happy to answer questions.